this is I'm going to talk about the chloroplast genome, but more about how we can manipulate chloroplast DNA. So, sorry. So as already I introduced this, I'm mainly focused on the chloroplast of green alga, Chlamydomonas langhardia, because this organism is amenable to a chloroplast transformation. So this organism is not only chloroplast but also nuclear, and as well as mitochondrial genome can be transformed. So I just remind of you the structure of this cell. So single cell, nucleus, and the chloroplast. And something like this. So you can see this is on microscopy. Looks like the flagella is moving around, and then chloroplast is. Is present in the stroma of the chloroplast, forming a nucleoid complex with DNA and the proteins, and some are loosely or so this is a circular DNA of the chloroplast genome, about 5,535 base pair, and here and here both the same DNA sequence repeated, duplicated, but in the opposite direction. Okay, and this is a small single copy region and large single copy region. Green part is exon, and the gray part is introns. Some chloroplast gene has introns, and also you may not see some genes. This light green is photosystem 1 encoded. Gene is encode a photosystem 2 subunits, and also blue is encode a subunit of cytochrome B6F subunits, and this yellow is genes encoding subunit of ATP synthase. Some subunits are encoded by chloroplast, and the subunits encoded by chloroplast is very often here the very essential subunits containing catalytic sites. And also this orange is gene expression involved in gene expression, RNA polymerase, the ribosome proteins, and other genes are involved in some PS1 assembly or chlorophyll synthesis protocol, fluoride, oxidoreductase, something. present in the chloroplast, and maybe its total number is usually less than 100. And the chloroplast usually has, it is estimated that about 3,000 or 2,000, maybe 3,000 proteins are present in the chloroplast. So only a small number of proteins are encoded by chloroplast gene. But for the photosynthesis reaction, the chloroplast encoded proteins, there are many very important proteins as a function. So I just today introduce chloroplast transformation. So as you can see here, the chloroplast is inside the cell, surrounded by two layers, cell wall and cell membranes, cytoplasmic membranes, so very hard. And the chloroplast is surrounded by two membranes, outer envelope membrane and inner envelope membrane. So DNA is here. So if you want to transform the chloroplast DNA, you have to deliver the DNA into the chloroplast. But there are at least four layers that prevent the introduction of DNA from the outside. So what we use is a particle gun to deliver DNA into chloroplast. So the chlamydomonas has the diameter is about 10 micrometers, and we use a tungsten particle with a diameter of about 1 micrometer, much smaller. And then we coat this particle with transforming vector DNA. Then shoot the chlamydomonas cell with particle gun at the speed of like a sound, 300 to 400 meters per second, very fast. Then this particle may block the cells like this, make a big hole, and then the cell will die. And maybe some cells may not be hit by particle. And fortunately, or I don't know, unfortunately for chlamydia, tungsten particle it transferred into the chloroplast, and the tungsten particle remain inside. Then the DNA coated on the surface of the particle will dissolve into the chloroplast, and then you can introduce the transforming vector into the chloroplast. So this is the particle gun we are using, and this is a controller, and this is a chamber. We make this inside the chamber, the vacuum. Okay, and then particles are shoot using the power of helium gas. Okay, very short pulse, helium gas pulse, shoot. And then inside on the agar plate, chlamydomonas cells spread on surface or agar plate with a monolayer. And then if the inside of chamber is vacuum, the small particle will not be accelerated so much, so very at high speed. You can shoot the cells with this particles. Okay, so this process is not difficult; it's easy. So this is how it works. So we on the agar plate, we spread cells like this. Basically, if you use a fixed concentration of cell and also you add optimize the volume, it's not difficult to spread the cells as a monolayer on the agar plate. And then you shoot the particle. Then we have a selection system. No, just after shooting, and then 1 week or 2 weeks later, you will see colonies like this. These are transformed. In the middle of the particle, the strong gas spread, blow away the cells and kill. So there was no, there's no transformed colony. So in the surrounding region, the optimized condition, you can see many colonies. The question is how you select these transformed cells. Is that we use a selector marker cassette that confer the cells antibiotic resistance. So we use the bacteria AADA gene that encode the enzyme that inactivate the spectromycin. That is, spectromycin is specifically block the protein translation in the ribosome of the bacteria type ribosome, not nuclear eukaryotic type ribosome. That is ATS. But this blocks the bacteria type 70S ribosome activity. So chloroplast has a ribosome of bacteria type ribosome of 70S. So this selector marker blocks the translation in the chloroplast, but with this selector marker, this inhibition, the block is removed. Okay, so with this,
we will add that antibiotics after the gene run, right?
Yes. Okay, good question. We already add the spectromycin on this plate, or after transformation, after shooting, we add the spectromycin. So answer is we use the agar plate with spectromycin. But we plate this, and within 1 hour, we shoot. Okay? So this short period, because we choose the concentration of spectromycin properly, so it does not immediately kill cells. Okay? So it's easy. No, you just shoot and put it on the incubator, in the incubator, and then you don't need care. Just 1 week or 2 weeks later, you come back and then see, oh, there are many colonies. So then you will be happy. So AD cassette, the bacteria type, is also a codon usage optimized for the bacteria, isolated this gene. But in this case, we can use AD cassette of which codon usage is optimized to chloroplast or Chlamydomonas chloroplast. And then only coding region will not be expressed in the chloroplast. This AAD openly different coding sequence is fused with 5' ATPA gene. It's a regulate 5' region of ATPA, chloroplast ATPA gene. And also, this is fused the 3' RBCL genes. Okay, so this region is very essential to regulate transcription and translation in the chloroplast. And this region is also very important for stability of messenger RNA in the chloroplast. But this region can be deleted. We know that it's possible, but because we don't know the efficiency may decrease or not, increase, probably decrease. So usually we use this. But in a specific reason, we don't want to use this 3' RBCL. So then we can remove this part. Okay, this part is not essential. So this is the first example of the heterogeneous expression, foreign gene expression in the chloroplast, and it worked. Okay. And then what happened if you introduce a transforming vector into the chloroplast? This is an example of targeted knockout of the chloroplast gene. So this is a host chloroplast DNA, okay? And this is a transforming vector, clone in the plasmid. And we clone first this target DNA. This is a target DNA we want to of interest. The upstream and downstream region, some region about 0.5 kb or 1 kb, certain size. We subclone this DNA into the plasmid. And then we insert in the middle of the gene AD cassette, spectromycin AD cassette. So that gives us a spectromycin resistance, okay, the white part. And then we introduce this transforming vector. So this homologous region, the red region, and blue region, the crossing occurs. So then this was replaced by this region in the transforming vector. So after transformation, AD cassette was introduced into this gene in the transformant chloroplast DNA. This reaction is enhanced in the presence of spectromycin. In the absence of spectromycin, this homologous recombination does not need to occur in the chloroplast. But in the selection condition in the presence of spectromycin, this recombination is enhanced. Otherwise, cell dies. Okay. So then this is insertional inactivation of the gene. You can knock out a gene by insertional inactivation. And or you can, if you knock out the gene of your interest, or you subclone the gene, and then you delete, or almost all the part or part of the gene coding region or all region. Okay, and then after deletion, you insert the AD cassette. Okay, like this. But between this homologous recombination, this happened. It's deletion mutant. This is insertional. This is deletion. But it contains AD cassette. Otherwise, this does not occur completely. Okay. This is mainly, you can knock out the chloroplast gene. But there's some gene that is essential for the survival of the chlamydomonas. So you cannot knock out. For example, the gene involved in the gene expression of chloroplast gene expression, you cannot knock out. There's some limitation. But photosynthesis gene can be easily knocked out because chlamydomonas can grow heterotrophically in the presence of acetate. Okay.
So CRISPR we cannot use in CRISPR-Cas we cannot use for chloroplast genome.
Sorry?
CRISPR-Cas
no, you don't need. This is easy. Nuclear transformation you can use, but this is you don't need at all. Very specific because homologous recombination occur like a bacteria in the chloroplast.
Sir, which vector is specifically using for this? Which vector is using? Bac vector or yak? Bacterial vector or?
Bacteria?
Which vector is using specifically any?
What the vector used?
Vector, but not bacteria. No, anything.
Anything you can.
Yeah. Blue script or no, PAC or PAC is not very important. But for cloning, the high copy number plasmid is very easy to. So usually we use the blue script for this. So then another the method is to site-directed methylation. You want to introduce a point mutation to change the codon or add the amino acid sequence into this. Like if you fuse the tag to the protein of your interest. Or you can delete some specific residues, something like that you can do, or insert in the middle of protein. So in that case, the transforming vector you subclone the same way, and this is a gene of your interest, and then you insert AD cassette downstream or upstream of the gene of your interest. And the position of the insertion does not affect the expression of this gene. No. Okay. Otherwise, the insertion of the AD cassette decrease the expression of this gene, so that affect accumulation of protein. So you need to find this position. The chloroplast. And sometimes there are many genes clustered. So you don't find the site to insert the selectable marker very close to the gene. So sometimes you have to insert it a little bit distant. So that happens. And also if you don't have any region, so then sometimes we can make two transforming vector. One is this selectable marker at the site, as already you know, that does not affect any gene expression, and also vector that has a point mutation. And these two transforming vector mixed and then transform together. So co-transformation, we call it. Then you select first the resistance against the spectromycin. Among the transformants growing in the presence of spectromycin, you screen the clone that has point mutation. So this is you need a little bit more work. And it depends on the experiment, but sometimes you have a positive clone that has a point mutation in, I don't know, 50% or sometimes 10%. But you can screen the tens of colonies, right? Tens of, 10, 20, 30, 40, 50, still possible. Then this, you need a longer homologous sequence here to insert. So the homologous recombination occurs, then both genes, mutated gene and the cassette are introduced into transformer chloroplast DNA. So this is a way. Because then before I talked about the insertion of tag to the chloroplast encoded protein. So this insertion was used this method. So I'm going to show our first knockout of gene, chloroplast gene, PSHC gene that is encoding iron sulfur protein of photosystem 1. This is one of the first chloroplast gene knockout. So PSHC gene is located here, single copy region. Okay. Yeah, this is the same thing. Okay. So we cloned this subclone, the DNA fragment containing a PSHC. So at that time, for example, it's a little bit technical. This is large EcoR1 fragment, and then subcloned PST1 and SAR1. Okay. This is an old experiment. It's published in 1991. So at that time, not so many technique. PCR was just commonly used. So to do this, by chance, this PSHC gene is small gene, but we found that NCO1 site is there in the middle of PSHC gene. And NCO1 site is here also. So we clone the PST1 and SAR1 site, or XBA1 and SAR1 site. Then there is no only NCO1 in the middle of the PSHC gene is a unique site. Okay, so we clone it into plasmid, PST1, SAR1, or XBA1 and SAR1, and then digest this plasmid with NCO1 restriction enzyme, and then insert it AD cassette here, ligate it. So the making plasmid was relatively easy. Okay. At that time, I was first engaged in molecular biology. So for me, it was a little bit difficult, but relatively easy now. It's very easy now. At that time, restriction enzyme was not so good, and then some enzyme didn't digest the DNA so well. That kind of things happened. Okay, then we transformed this plasmid into chloroplast. And then we checked the transformant. Selected the cassette spectromycin in the presence of spectromycin, and then putative transformant. Until we confirm the DNA, we can't say it's a transformant, because sometimes a spontaneous mutation becomes spectromycin resistance. Okay. Then at that time, now we use a PCR, but at that time we used the Southern blotting experiment. Do you know the Southern blotting? Yes? Recently, only few people use Southern blotting. But for now to analyze this, the transformant, just we need to amplify near PSHC, and then if there is an insertion, PCR product becomes larger. Without the insertion, PCR product is like wild type. It's easy. But at that time, we have to analyze the Southern blot analysis only. PCR reaction was sometimes very tricky at that time, so and also expensive. So the oligonucleotide synthesis was also not cheap. So we used Southern blotting. So we digested total DNA with what is EcoR1 and SBA1, so EcoR1, XBA1 here and here, from here to here, okay? Here to here. And then separated a fragment of DNA by agarose gel electrophoresis, and then blot it onto the membrane, and hybridize with a probe of this region, okay, with labeled with phosphate. And then we detected by X-ray film, and then developed, and then the fragment hybridized with radioactive probe was detected. Okay, this is Southern blotting. And the wild type here, its size is 3.0 kilobase pair, KBP. But if PSAC has an insertion of 1.9 K kilobase pair cassette, so the size increases to 4.99 kilobase pair. Okay, don't worry about this additional one. So at that time, somehow something happened. So as you expected, this is a probe with PSAC gene. So 3 kb signal are upshifted for 4.9. So it shows all DNA are transformed. Chloroplast DNA are transformed. Okay. And we also hybridized with this AD cassette. The probe hybridized with AD cassette. Then there's no signal in wild type here, but signal at 4.9 kilobase pair appear because this contains PSAC and ADA gene. So we confirmed that, oh, we can make. We made it. Okay. Then what we will do is analyze the phenotype of transformers. We did another analysis, the messenger RNA level. So in the wild type, the messenger RNA, the probed with PSAC gene, give us a 0.45 kilobase messenger RNA and 1.1 kilobase messenger RNA, 2 messenger RNA. But in the transformant, we couldn't detect any transformant, so transcription didn't work or very unstable. The messenger RNA becomes very unstable. So the AD cassette messenger RNA, there is no in the wild type, but in the mutant, there's signal. So AD cassette is transcribed and probably translated because the cells are resistant against spectromycin. Okay, so today I'm not going to talk about these two, but just I tell you that we did mapping. So 0.5 is starting here and then end in the downstream of PSAC. So this transcribe only PSAC gene, and larger one is the starting is the same, but it goes through a little bit downstream. And then we found the open ready frame downstream of PSAC at that time. Then that was encoding photosynthesis protein we found at that time. Whole genome of chloroplasts was not have not been determined. So maybe if I come later, sometimes I talk about this. So the phenotype, another phenotype, the activity of the transformant. We measured the fluorescence induction kinetics. I think you did some laboratory course this time, PAM measurement, okay? So this is a chlamydomonas fluorescence induction kinetics. It's time about 2 seconds. Then you illuminate the cells with actinic light, fluorescence go up a little bit, how do you say? Strange kinetics and the peak, and then go down. Okay, this is Y type. Without killing the cell, breaking the cell, you can measure this kinetics. And 4B2 is one of the PSAC knockout mute transformant, one of the clone. Okay, so kinetics like this, it's exponentially go up and then stay constant. It's completely different. So you know from your laboratory course that what this phenotype means. Did you try, experience something like this? Tell me.
Speak loudly.
You want to speak microphone?
What did you ask? We are asking what did you ask?
What did you ask?
Can you repeat the question?
This is Y type kinetics, and this is transformant. So but this difference in the kinetics of fluorescence induction kinetics, what this means? Absence?
Fluorescence in mutant is more than wild type.
Sorry, I don't hear well.
Fluorescence in mutant is more than wild type.
Higher. Yeah, obvious. But what this suggests? You can explain why it's higher. Did you do this kind of experiment? Yes. Yes? Okay, why is higher? So, okay. During illumination, the fluorescence yield increases like exponentially. This means photosystem 2 activity is there. Okay? So it goes higher and it didn't go down. Okay? So this means that the fluorescence is basically emitted from photosystem 2. The photosystem 2 acceptor QA or praskinon pool is reduced, fluorescence yield increases. And its QA or praskinon pool is oxidized, fluorescence yield decreases. Okay, so this means wild type, it's very complicated kinetics. PS2 is reduced, but certain point it starts to oxidize because photosystem 1 extract electron from PS2. So oxidize praskinon pool and oxidize acceptor of PS2. So this means PS2 activities here in the wild type and also PS1 activities here. But in the mutant, it's go up. So there's no activity to extract the electron from PS2 to from PS2 because on the PS1 activity is absent. So electrons accumulate. So PS2 is highly reduced. So the fluorescence yield becomes highest. So it happens higher, and they didn't decline. So this indicates that PS1 activity in this mutant is absent. This is as expected because PSSC encode iron sulfur protein of photosystem 2. This is essential for PS1. So this is as expected. But we can confirm easily by measuring fluorescence induction, the phenotype. Then we expect the PS1 protein is absent or present, but not functional or non-functional. So we just simply analyze the protein polypeptide of thylakoid membrane. We isolate thylakoid membrane and analyze the polypeptide pattern. This is wild type. PS1 reaction center is around here, and some proteins are PS1. Okay, there are many. And number two is negative control of the mutant. Already we know the PS1 is absent.
The acceptor side is there, so the oxidation is affected.
Yeah.
So, but why reduction is also maximal in mutant?
This is the balance between the reduction and oxidation. Okay, and also it's always this is also balance of redox between the PS2 and the PS1. So there's some explanation, but I'm not quite sure if they are correct or not. But this is also reflect because PS1 and PS2 activities start at the same time if you put on the light. So always, the equilibrium occurs. So in the absence of PS1, this kinetics is totally absent. But in the presence of PS1, there's some the balance is.
The energy distribution will be
energy distribution or the balance of the distribution of electron among between the PS2 and the PS1. But I'm not quite sure. There's some explanations, but it's correct or not. Sometimes I doubt some explanation. But this kinetics is always observed in the wild type, but even the PS1 and the B6F deficient mutant is same like this. This disappears. So this is always appears in the presence of PS1 activity. So PS1 in transformers almost absent here. Okay. So although we knocked out the PSAC genes that encoding a small subunit of PS1, but other PS1 protein were also seem to be absent or decreased. Then we tested reactions, the immunoblotting analysis. At that time, the sensitivity of immunoblotting was not so high because we didn't have the technique of enhanced chemiluminescence ECL method. At that time, ECL was not, we didn't know. We still had, how to say, this was used isotope to detect high sensitivity. It was a little bit scary because iodine 125 is very strong gamma ray, so I have to work in lead metal, so heavy. So if you use the radioisotope emitting beta ray, it's okay, but the gamma ray is very powerful, so it penetrates. So if you want to shield yourself, you need a heavy metal block. So it's not easy to work. And PS1 deficient mutant completely lacks PS1 reaction center protein, but it a little bit accumulate in the PSAC mutant. It's very much reduced. So immunoblotting is more sensitive. So other small subunit is totally absent. So the question arises why PS1 is other PS1 subunits are reduced in the absence of PSAC. This means that the knockout in PSAC gene or translation affects the translation of other PS1 subunits, block or suppress the translation, or synthesized PS1 proteins unstable and degraded. So to analyze this, we did a pulse chase labeling experiment as I talked before. Okay, we used this S35, I think, at the time. So we labeled, and 2, 5, 8 is the labeling, pulse labeling, and 3 and 4 is chase experiment. So this was reaction center protein, and the wild type pulse labeled protein was stable during chase experiment. Okay, and this PS1, no, not really. This is only chloroplast encoded protein. And PS1 minus mutant is also pulse labeled. It's a little bit strange, but these are not really PS1 because it's upshifted. And PSAC mutant is pulse labeled like this, and during a chase it disappeared. So this means if you knock out PSAC, reaction center protein, other PS1 subunit, the main reaction center protein, synthesized but degraded rapidly. So if the synthesis rate is the same but degradation rate is faster, so steady state level of accumulation of PS1 is reduced like this. Maybe 20% or 10% of wild type level. So this means that PS1 complex is unstable in the absence of PSAC. Okay, this is a example of knockout of chloroplast gene. So this is first experiment for. So we just knock out already function the gene of which product function is known. Okay, because we needed to test the phenotype. So without unknown, if you work with the gene that is of which function is unknown, in the beginning it was not easy to analyze the phenotype, so we choose this gene as a target. Okay. So I think next is the point mutation, but its time is running out, so I think that tomorrow I will talk about introducing a point mutation. And so I will stop this talk. So I welcome your question and comment or anything.